Hey everyone. Today we're exploring how cells manage to transport materials in and out across their cell membrane. Picture the cell membrane as a security gate, constantly deciding who gets in and who stays out. But what makes this gate so unique is how selective it is. Not just anything can pass through. The cell membrane, also called the plasma membrane, is made up of a double layer of lipids with proteins and carbohydrates embedded within it. This barrier acts as a selective guard, allowing certain molecules to pass through while blocking others. So, how does the membrane decide who's allowed through? And some molecules can simply walk right through, like they have a VIP pass. Small nonpolar molecules such as oxygen and carbon dioxide diffuse rapidly through the cell membrane. Small polar molecules like water can cross as well, but they do it at a much slower pace. On the other hand, larger molecules, whether they're nonpolar like vitamin A or polar like glucose, find it nearly impossible to get across without help. And then, we have highly charged ions like sodium or potassium. They don't stand a chance of slipping through on their own. Now, just like a VIP guest who needs assistance to get past security, certain molecules rely on transport proteins to help them cross the cell membrane. There are two types of transport proteins that act as gatekeepers here. The first are channel proteins, which form tunnels that let specific molecules pass through. Aquaporins, for example, allow water molecules to flow in and out of the cell, making sure hydration stays in balance. The second type is carrier proteins, which act more like revolving doors, changing shape to move larger molecules like glucose across the membrane. But what happens when cells need to transport a large quantity of material or something too big for the usual channels and carriers? This is where the cell gets a bit more creative, using processes called endocytosis and exocytosis. Let's break them down. Endocytosis is like the cell reaching out to grab something from the outside and bringing it in. Imagine a white blood cell, like a macrophage, coming across a pesky bacterium. The bacterium attaches to receptors on the cell surface, and then the macrophage extends its membrane around it, like giving the bacterium a giant hug. The cell membrane then closes around the bacterium, forming a vesicle called a phagosome. This vesicle floats through the cell, eventually meeting up with a lysosome, which is packed with enzymes ready to break down the bacterium. This is a classic example of phagocytosis, one type of endocytosis. But there's another type called pinocytosis, where the cell drinks extracellular fluid, taking in whatever solids happen to be in the fluid. It's less selective and more about grabbing a quick sip. There's also a third type called receptor-mediated endocytosis, which is a more selective way of bringing in materials. In this process, specific molecules like LDL, the low-density lipoprotein, bind to receptors on the cell membrane. Once bound, these receptors gather into pits coated with a protein called clathrin. As the pit deepens, it pinches off, forming a vesicle that enters the cell. Inside, this vesicle eventually separates, and the LDL is sent to a lysosome, while the receptors are recycled back to the cell membrane, ready to do their job all over again. Now, sometimes the cell needs to export materials, and that's where exocytosis comes into play. This process begins deep inside the cell, with an organelle called the Golgi apparatus. The Golgi takes proteins, lipids, or hormones, and packages them into a vesicle. The vesicle then travels along the cell's cytoskeleton, guided by motor proteins like kinesin or dynein, until it reaches the cell membrane. Once there, the vesicle fuses with the membrane, and its contents are released into the extracellular space, like a cell delivering a package to the outside world. Both endocytosis and exocytosis require energy in the form of ATP. This energy is what powers the movement of vesicles, the work of motor proteins, and the fusion with the cell membrane. It's like fueling up a car before a long road trip. Without ATP, these processes simply wouldn't happen. To sum it all up, cells are constantly busy transporting materials across their membranes. Small nonpolar molecules can pass through easily, while larger or charged molecules rely on transport proteins. When it comes to moving a lot of material or bigger items, the cell turns to endocytosis to bring things in and exocytosis to send things out. And behind every move, there's ATP powering the whole process. So the next time you think about cells, remember they're not just sitting around, they're actively managing their own little world. Thanks for joining me on this journey through the cell membrane. If you're looking to dive even deeper into fascinating topics like this, be sure to check out King of the Curve. We have a wealth of resources to help you excel in your studies and make learning fun. Don't forget to like this video, subscribe to our channel, and hit that notification bell so you never miss an update. Until next time, keep exploring the incredible world of biology.